So this is Andres Rivera-Quiroz, and he'll be telling us today about pushing the limits of micro-CT visualizations, PTA staining to observe the spider central nervous system.
Thanks, Brian. I'm back, by the way. So.
Oh, okay, Mike.
We'll see how that goes.
Okay.
Go ahead.
Well, should I go ahead?
Yes, absolutely. Sorry.
Okay. Well, yeah. Thank you for the introduction, Brian. I'm gonna show you here the results of a first step in a series of studies that Jeremy and I have been doing about the spider brain, and it's a new staining protocol that allows visualizing the whole internal anatomy of the spider, but that we have been using for observing and characterizing the central nervous system, and specifically the brain and the visual pathways. So, yeah, I want to start just by acknowledging how important proper morphological and anatomical documentation is to better transmit our ideas and test our ideas and hypotheses about taxonomic relationships and character evolution. And well, just to name a few here, we can start with scientific illustration, stacking images, histological preparations, and of course, more modern techniques like electron microscopy. But those, or most of the ones I mentioned, work perfectly for documenting the external morphology. But the internal anatomy, it's always a bit more difficult because, for one, it's inside the specimen, and also it requires a series of steps for staining, fixating, and also eventually slicing the specimen, which also means that the specimen is in the end destroyed. These techniques allow characterizing the structures in a finer detail up to a cellular level, and are really, really important to know where everything is connected to. But for doing all of these characterization and visualizations without actually destroying the specimen, micro-CT has been suggested in a study a few years back using insects, and was tested to visualize the neuroarchitecture of the insects. And then a lot of studies on the spider neuroanatomy followed that same protocol. The only problem with this protocol is that it requires the specimens to be critical point right to achieve the maximum resolution and the best contrast of the tissues. And this induces the tissue shrinking, and in many cases, the deformation of the internal organs. So having all of these problems in mind, it's not surprising that we don't actually know a lot about the brain in an evolutionary context. We know a lot about some model organisms, but there are actually only a handful of studies that have used broad taxonomic sampling. One of those, a very recent study, tried to categorize this variation in relation to the time of activity and the type of hunting that the spiders display. So what we want to do with our project overall is to get a better understanding of how the brain works and how it is how it varies from one group to the other, and then relate it to other phenomena like eye reduction. But of course, for achieving all of this, we first need to develop a visualization method based on micro-CT scan that is fast and therefore allows to analyze big number of specimens. It gives us enough resolution to differentiate and identify tissues and structures, and also that does not require critical point drying because of two main points. One, that it avoids the creation of artifacts, and also because it would allow to use legacy material from collections, giving us the opportunity to broaden our taxonomic sampling and also to represent rare species. So we tested first the application of the protocol I mentioned before, based on iodine, in a group, well, on a series of Araneus diadematus specimens. And here we can see on the left side, the iodine staining with the specimen in ethanol, and on the right side, the specimen after it was critical bond dried. And you can see that indeed there's a much clearer image, but there's a high reduction and deformation in some tissues like the muscles and the central nervous system, digestive system, among many others. And so for overcoming this problem, one of our technicians suggested the use of phosphotungstic acid, or PTA, that has been used recently in other marine invertebrates, some plants, and even mammal embryos, and has some advantages similarly to iodine, that is not toxic and it can be diluted in high concentration of alcohol. And but in contrast to iodine, it gives us a great contrast of the tissues without the need of critical point drying. So we went ahead and used also Araneus diadematus specimens to test the applications. And then we hit the first and only wall so far with the application of PTA, and that is that it takes too long to actually stain the specimens. On the far right, on the first left, you can see a specimen is stained with iodine after only 1 day. And on the far right, you can see a specimen stained with PTA after 25 days. However, we found this suboptimal stained specimen after 10 days that allows us to clearly visualize the brain and the central nervous system. Just to give a comparison of the quality of the image we can get with PTA in contrast to iodine, here I show a section of the whole cephalothorax of the spider, and also another section, a sagittal section of the brain, comparing both techniques. And also, by modifying some settings and a higher magnification in the scan, we were able to observe very tiny structures like the retina in the different eyes and the optic nerves, and also to recognize the brain pathways. In this case, I'm showing the secondary visual pathway in the brain. But we were still not happy with actually having to wait between 10 and 25 days. So we tested if we could improve the times of the penetration in the tissue by the PTA by applying vacuum to the samples. And we did this in two different ways. One with an intermittent vacuum, where the samples were put in PTA and left in vacuum for 10 minutes, and then left in a rotator for 24 hours. And a continuous vacuum approach, where we put the samples in PTA and left them in the vacuum, in a constant vacuum for the whole time that they were being stained. So in the end, we found that this sweet spot that I talked about was achieved in constant vacuum after only 2 days, and in intermittent vacuum after 5 days, effectively cutting the staining times by 50 to 80%. And the best part is that although we can see some differences in the quality of the scans, we were still able to identify all the parts of the brain and recognize them. And even we were able to make 3D reconstructions of all of these parts. We focused especially on the optic nerves, on the optic neuropiles, and the whole central nervous system. So with this, we can conclude that PTA indeed provides a much better contrast than the commonly used iodine staining, that it allows the visualization, identification, and reconstruction of the internal anatomy, in particular for us, for the visual pathways. But it does so avoiding critical point drying and the shrinking and the formation of the soft tissue that comes with it. And also, of course, it has a lot of potential applications in other systems and tissues, such as respiratory systems, digestive systems, venom glands, among many others. And well, we are currently applying this protocol, and we will be doing so in other projects for contrasting the quality of the scans that we can get from freshly collected specimens versus legacy material. And here we want to assess the impact of ethanol preservation on the neuroarchitecture. And also we want to quantitatively analyze the intraspecific variation by reconstructing and comparing the volumes of the brains and the different neuropiles, and also by applying a 2D geometric morphometric approach. And we also want to do a multi-taxon approach to study the evolution of the optic nerves and visual pathways between different spider groups. And we want to do so by also by quantitatively analyzing the interspecific variation. But in this case, we want to do 3D morphometrics for analyzing the shape of the optic nerves in the different groups. And finally, we will take all the lessons we learned from that to observe the evolution and the plasticity of the visual pathways in relation to eye reduction and eye loss in spiders, especially from caves. So with this, I would like to end my presentation and well, thank Naturalis Biodiversity Center for funding me. And thanks to my supervisor, Jeremy Miller, my group leader, and the technicians that have helped a lot in developing this protocol. And I'll be happy to take your questions now.
Wow, great job. That was amazing visualizations. I do have a little bit of time for questions. Hannah Wood has a question. She says she's surprised that there were problems with iodine. Were you using Lugol solution? Did you wash the specimen for a longish time after staining with iodine?
Yeah, we didn't use the Lugol solution. Yeah, that might have been a problem from our protocol. But although the level of shrinking that we had is a bit higher than what is reported in literature, still, if we avoid that step of drying the specimens, we can completely avoid the shrinking whatsoever. So, yeah.
Yes, great. And then Brian Patrick just wants you to explain, how does critical point drying introduce artifacts?
Yes, when the tissue shrinks, it can deform the organs, and this is especially seen in the softest tissue, like the central nervous system that I was looking at, and also in the musculature. And yeah, so the shape that you are looking at in the reconstruction is not the real shape that you would see in situ if the specimen wasn't dried. So that's the kind of artifacts that it can create.
Thank you, Andres. I think